Here. Hi, good morning. I'm Sae Tanaka from Keio University. So I'd like to talk about the analysis of the glucose solution using near-infrared spectroscopy. We love sugar because sugar is essential for a living organism. It gives us not only sweetness, but also energy for a cell to work. When sugar bonds each other or other molecule like proteins, it can make cell structure and or the work as the molecular recognition part. In living organisms, sugar is used everywhere in various forms. Glucose is the most abundant sugar molecule in living organisms and in the world. Glucose consists of the 6 carbon, 12 hydrogen, and 6 oxygens. The object of this study is to understand the interaction between sugars and water. At first, I imagined the sugar solution like this, but sugar solution is not so simple. So, in the fact, glucose has 2 isomers. They are called anomers. In solid states, like a powder or a crystal, alpha glucose is measured. On the other hand, in aqueous solution, the beta is measured form in the solution. They are balanced at the ratio of 4 to 7 in the solution. It's because a hydrogen bond between sugar and the water molecule are formed around the C1 carbon. Therefore, to analyze glucose solution, first of all, we have to distinguish these anomers in near-infrared spectra. So, near-infrared spectroscopy can detect the molecular vibration such as CH or OH. This chart shows the absorption of each molecular vibration in the near-infrared wavelength region. Sugar has CH and OH, and water has OH. So we mainly analyze from 1100 years to 1800 here in this study. Here, today I talked about difference between glucose anomer spectra in analysis with concentration and in the ratio of anomer in solution. And then I'm talking about the origin of the near-infrared band in the analysis using deuterated glucose. First of all, we got both pure anomer powder and prepared each solution, and we measured each anomer solution from 25 to 200 millimolar to examine the difference between the anomers. So I show here the subtraction of the glucose beta glucose anomer solution spectra from the alpha glucose spectra. So you can see the clear difference here at the 17. You can see the 1742 nanometer. It also shows the clear concentration dependency is here. And in addition, there are many bonds. There are several bonds in the OH region and the CH region. Although the concentration dependency is not so clear. So next, we performed the PLS regression analysis on the same data to evaluate the concentration dependency statistically. So I show here the regression vector that show the contribution on making the linear model of concentration. Regression vector of the alpha glucose is indicated by the blue line, and the beta glucose is indicated by the orange line, and the difference between them is indicated by black line here. So here you can see the significant difference in the CH and OH regions. Compared with the earlier result of subtraction, the peaks at 11, 18, 98 nanometer and 1742 nanometer appeared in both charts. As I mentioned earlier, glucose anomers are balanced in aqueous solution, but it takes about 2 hours from dissolving to balanced state. Therefore, next, we examine the spectral change with time. To know the ratio of anomer in the measurement solution, we use a polarimeter. Specific rotation is calculated by this formula. Chemical compound has characteristic value of the specific rotation. For example, alpha glucose is 112, the beta glucose is 19, and the equilibrated solution of the glucose is 15.3. So thus, we can calculate the ratio of anomer from the specific rotation. I show here the time course of the measurement. 200 millimolar glucose solution were prepared with each anomer powder, and the solution of 5, 13, 1 hour, 2 hours, 3 hours after dissolving were measured with near-infrared spectroscopy and the polarimeter. In the PLS regulation analysis, regulation model of anomer ratio was vetted using near-infrared spectra and specific rotation value. In the left panel, the major value and predicted specific rotation value are plotted. It shows a linear correlation. And in the right panel, I show the regression vector of the specific rotation. The peak at 1742 nanometer appear again here. And it is the highest peak in the regression vector. Therefore, we conclude the absorbance at 1742 nanometer is the most important to distinguish the glucose anomers. Next, we examine whether a single absorbance at 1742 nanometer show a correlation with specific rotation. There was some baseline shifted due to the temperature change, so we normalize the spectra using line subtraction. As a result, the absorbance at 1742 nanometer show the strong correlation with the specific rotation. In addition, to examine whether the correlation between absorbance at 1742 nanometer and specific rotation in biological condition. We measure the glucose anomer solution in PBS. PBS is phosphate buffer saline, and at 37 degrees, it's a body temperature. So I show here the both results. Upper panel is glucose anomer solution in PBS, and lower panel is a glucose solution at 37 degrees. You can see the strong correlation between absorbance at 42 nanometers and specific rotation remained in biological condition. It suggests that this wavelength is useful to predict the anomer ratio of glucose in cell or bodies. Next, we measured the Maltose solution to examine whether this prediction method using absorbance at 1742 nanometer can adapt to the disaccharide. Maltose consists of 2 glucose, and it has also 2 anomers. They are balanced at the ratio of 3 to 7 in the solution. So absorbance at 7042 nanometer also show the strong correlation with the specific rotation in maltose solution. It suggests that this absorption is highly correlated with the structure change at the C1 carbon. But there are several peaks in the regulation vector of glucose anomer ratio here. So based on conventional assignment, these peaks should be originated from CH, or these are from the OH or CH. There are obvious difference in the structure on the C1 carbon. So the interpretation that these peaks originated from C1 carbon may be reasonable. But actually, there are several reports about structural change between glucose anomer, for example, on the exocyclic CH2. So at first sight, I guess these bands originated from the exocyclic CH2 because this spectral pattern is similar to the CH2 asymmetric and symmetric band. Yeah, but what is the origin of these peaks? I think we have to examine the origin of these peaks based on the experimental data. So to assign each peaks in regulation vector, we are now trying to analyze deuterated glucose solution. Deuterium has 2 times weight of hydrogen, so exchange to deuterium causes to peak shifts like this. This is a powerful tool for band assignment in vibrational spectroscopy. Here we use 2 types of deuterated glucose, glucose 1D1 and glucose 662. We measured deuterated glucose solution and performed the PLS regression analysis. I show here the regression vector of the specific rotation in CH region. Glucose 1D1 spectra shows a difference pattern in the regulation vector compared with glucose and glucose 662. It suggests that absorbance at 1742 nanometer, that absorbance at 1742 nanometer is mainly originated from the C1 carbon. Finally, I want to touch the OH and the CH region around 4000 nanometer in the regulation vector. So in this region, there are possible peaks from CH and OH of glucose and OH of water molecule. You can see there are 2 peaks change to opposite in the glucose 1D1. Actually, although I thought these peaks originated from exocyclic CH2, as I mentioned earlier, so it's not correct because the peaks remain in the regulation vector of glucose 6,6-D2, so there are two peaks. So it is relevant to think these peaks are originated from OH of glucose or OH of water. But I think the further analysis is needed to conclude these peaks' origin. Yeah, so as summary, we analyze glucose anomer solution using near-infrared spectroscopy combined with polarimeter measurement. And so, and to assign the origin of band peaks, we use two types of deuterated glucose solution. As a result, we assigned CH band originated from CH on C1 carbon, and OH of OH band originated from water or glucose. We are going to perform in further analysis using deuterated glucose solution to understand glucose spectra. Thank you for attention.
Thank you very much. The floor is open for questions. Do we have questions? Questions? Yes, sir.
So, sugar and water form crystals. Right?
Yeah.
You can have even a solid crystal of sugar and water. Could you say something about what you think happens in a situation like that?
In the crystal?
What happens to the sugar in the water when the crystal forms? Do your results suggest something specific about that?
So in the crystal, the glucose is a form. This alpha of glucose is measured almost 19 or 80% is in the crystal. So the water, beta glucose has water in the here. There is a hydrogen bond with water and glucose in the beta glucose, but in the crystal, the water and there is no interaction with water and glucose. Pure glucose is more stable than the glucose with water. So this form is more stable with the water, like in the solution or it's in the solution. So my conclusion is cannot lead to some answers. Sorry.
Some other questions? Yes.
We know that sugar is one of the fundamental components of DNA. Can you mention about the role of the diluted sugar in the body water to maintain the stability of DNA? Because in the dynamic of biological processes, DNA needs to renew its structure. And what is the role of the body water and diluted sugar in the body water in the process of this process of recompensation of the losses of DNA in the cells?
Okay, so in the DNA, the different sugar is used. It's maybe 5 links sugar is used in the DNA. So it's a different story. But I think the sugar has a role in the keeping water in the body or cells. for example, we use a lotion, skin lotion. It contains some sugar. It is called trehalose. Trehalose has a strong correlation with dehydration and hydration. So I think this function, the structure of the glucose is very important. Because the sugar has a hydrogen bond with water, depend on the structure. So I think the beta glucose is more stable in the solution, but actually in our body use the alpha glucose more often. I don't know why, but I think it's a very interesting point when we studied on the glucose or sugar in the body or cells, I think. Yeah, thank you.
Thank you very much. We run out of time. Thank you very much. Our next speaker